Good day, we are Group 8 and we're going to present Activity 6 about the restriction enzyme digestion and ligation. So restriction enzyme digestion and ligation of ligase is important in genetic engineering, where these two techniques are usually used to transfer a DNA fragment to another DNA. If you recall, in recombinant DNA technology, the DNA fragment that is transferred is called the gene of interest, while the DNA where it is inserted on is called the vector. So with restriction enzyme digestion, it allows us to cut out and isolate the gene of interest, and with ligation, it will allow us to combine the gene of interest to the vector, making the recombinant DNA. But aside from making recombinant DNAs, there are also many more uses of digestion and ligation, such as in constructing a DNA map or for site-directed mutagenesis. Essentially, this topic is just about the cutting and gluing of DNAs using these enzymes we found from bacteria. The two main enzymes that drives DNA digestion and ligation are restriction enzymes and ligase. But in this video, we're just going to focus more on restriction enzymes. And basically, the only thing that you need to know about ligase is that they're the ones that catalyze the ligation or the joining of two DNA fragments. And you pretty much already heard of them in the DNA replication of the lagging strand or in DNA repair. So restriction enzymes, on the other hand, are the enzymes that cleaves the two strands of DNAs. So they are endonucleases, which cleaves at the centers of DNAs, unlike high-fidelity polymerase and other exonucleases, which cleaves at the end of DNAs. They also have DNA sequences that is there's also then DNA sequences that is recognized by the enzyme, which is called the restriction site, which are usually 4 to 8 nucleotides long and usually palindromic. So, for example, is BAM-H1, which has a sequence of GG80CC, and its reverse complement is the same, which is GG80TCC. Then these restriction enzymes also forms blunt or sticky ends. So for example, ALU1 forms blunt ends while BAM-H1 forms sticky ends. So this presentation will be divided into two parts. In the first part, we'll tackle all the activities related to digestion, while in the second part, we'll tackle the activities related to ligation. So for our objectives, first is we aim to learn how to use the two softwares, namely the Snap Gene Viewer and the APE. Then specifically, we want to be able to read a plasmid map using Snap Gene Viewer and to generate a virtual digest using APE, and also to construct primers, determine amplicon, and edit plasmids for in silico blunt and sticky end ligation.
To get started with the digestion part, we'll first start with learning about plasmids. Plasmids are small, circular, and self-replicating DNA that are separate from the cell's chromosomal DNA. Some of the notable characteristics of plasmids can be seen by generating a plasmid map like the figure on the right. We see that plasmids contain an origin of replication and a promoter region which drives transcription of the GOI. Plasmids also contain an antibiotic resistance gene and a selectable marker, both of which can be used for selection of cells, either separating those containing plasmids and those that do not, or those that contain the transformed plasmid with GOI. Now let's open a plasmid sequence using Snap Gene Viewer. First, we open the file sequence through the app. Once it is opened, we make sure that it is circular by looking at the top right icon. Then we go to the Features tab and click Detect Common Features. We can see the list of common features here and we click Add Features. Now we can see different parts added to our plasmid map. We can also see these in List View through the Features tab at the bottom. Our output shows the plasmid map with all the new features pointed out. Among them, we have the CMV enhancer and CMV promoter, where CMV stands for cytomegalovirus. This is a mammalian promoter that initiates transcription. Next, we have the SV40 promoter, another expression promoter, this time from the simian vacuolating virus. We also have the SV40 origin of replication. Moreover, we also see two poly-A signals, SV40 and HSV-TK, which play a role in the termination of transcription. Moving on, we see NeoR or CAN-R here, which serves as resistance to the antibiotics neomycin and kanamycin. As mentioned earlier, these help in selection as the bacteria is pressured to keep the plasmid for antibiotic resistance and could also help the researchers identify plasmid-containing bacteria. We now move on to learning about the multiple cloning site, or MCS, which contains multiple recognition sites of restriction enzymes. This can be seen in the plasmid PUC18 shown on this slide. The MCS allows different DNA fragments that are cut with various enzymes to be inserted, thus leading to great flexibility in the range of possible inserts. Going back to the plasmid we opened earlier, where does its MCS lie? We look for a region where there are many restriction sites, which is here. We go to the Sequence tab at the bottom to be more specific in selecting this site. Then we click on the Features tab at the top and click Add Feature. Renaming this to MCS, we also change the color of the feature. And now we can see the MCS in our plasmid map. Now that we've seen how to read a plasmid map, let's look at how to digest a plasmid with restriction enzymes. Let's say we have a plasmid with 3 restriction sites of a certain restriction enzyme. Upon digestion with this enzyme, the plasmid is cut into 3 sites, resulting in 3 fragments. We can use more than one restriction enzyme. In the case of using 2 simultaneously, we call it a double digestion. Let's try this once again on SnapGene. First, we go to the Enzymes tab and click Choose Enzymes. We remove all the enzymes present and add our target restriction enzymes, NDE1 and PST1. It says here that we should be expecting 2 restriction sites each for these enzymes. With these enzymes, we can determine the number of base pairs up to these cut sites. For example, with single digestion with NDE1, we see a cut site at 233 base pairs and at 1016 base pairs. While for PST1, we see one cut site at 603 base pairs and another at 3687 base pairs. Plotting these sequences on a plasmid map, we can calculate the size of the fragments upon single and double digestion. Our plasmid size is 4553 base pairs. With NDE1 single digestion, we expect 2 fragments. The purple one at 783 base pairs and the green one at 3770 base pairs. For single digestion with PST1, we expect to see the green fragment, which is 3084 base pairs, and the purple fragment, which is 1469 base pairs. If we perform double digestion, we see 4 fragments, one from A to B, B to C, C to D, and the last from D to A, with the fragment sizes shown on the slide. Incorporating these fragment sizes into AGE with a DNA ladder, we should expect the following fragments. Lane 1 here holds the Takara 200 base pair DNA ladder, Lane 2 is for single digestion with NDE1 only, Lane 3 is for PST1 only, and lastly, Lane 4 is for double digestion with both of the enzymes. The fragments were drawn by using the DNA ladder as a reference and estimation point. To check the AGE profile we came up with, let's compare it using the APE software. We first open the sequence file using APE, then make sure it is circular by checking the icon at the top right. Next, we go to the Enzymes tab at the top, Ladder, then Edit Ladders to manually input the ladder we used. A dialog box appears, and here we type in each DNA fragment in the ladder. It's already been added, but let's try to add the last 200 BP fragment. We hover over the fragments to check for accuracy. Now we select the enzymes NDE1 and PST1 through the same Enzymes tab. Click Highlight, and the cut sites should appear on the sequence. We also generate the graphic map through the same enzyme selector, and notice that it is similar to the one we drew earlier. We click on the Digestion dialog box under the same Enzymes tab to add the lanes, similar to the AGE profile we drew earlier, and click OK. We now have our AGE profile generated using APE. Here is a comparison of the APE-generated graphic map and the SnapGene graphic map. There are minor differences in the sequence numbers, which could be attributed to differences in algorithms and in setting the arbitrary 0 marker for the plasmid, as indicated by the stars. Results also showed that our drawing and the APE-generated AGE profile are quite similar. Moving on from how to digest a plasmid, what if we have a novel restriction enzyme? How can we determine the optimal conditions for it to work? Let's consider a certain restriction enzyme X extracted from a bacteria isolated from a sea sponge. Its 3D structure has already been elucidated, and analysis shows a possible 6 base pair restriction site. In this activity, we determine the optimum conditions of temperature, magnesium ion concentration, and incubation time. We do this by performing AGE with the pure undigested plasmid, as well as the digested plasmid at different conditions to see which condition yielded a complete digestion. This is followed by the determination of its RE site through the digestion of a standard 1 kb DNA sequence. First, this AGE profile shows the digestion of the plasmid with REX at different temperatures. The highlighted lane 2 contains only the plasmid, and lanes 3 to 7 have increasing temperatures. The optimal temperature should result in successful digestion of the plasmid into smaller fragments. Looking at lanes 3 to 7, we see that lane 4 successfully digested the plasmid, as the 2700 base pair of the pure plasmid is not present, and there are 3 smaller fragments observed. Thus, the optimum temperature is 30 degrees Celsius. Second, we performed the same approach in determining the optimum concentration of magnesium ions. Lane 2 contains only the plasmid, while lanes 3 to 7 have varying concentrations from 0.1 to 0.5 millimolar. Again, the optimum concentration is determined by the disappearance of the band corresponding to the undigested plasmid and the presence of smaller fragments. This is seen best on lane 5 compared to the other lanes, although this lane has remnants of undigested plasmid fragments. Still, it is the optimum concentration among the 5 concentrations. Hence, 0.3 millimolar is the optimum magnesium ion concentration. Hence, 0.3 millimolar is the optimum magnesium ion concentration for the activity of REX. Third, based on this AGE profile, the optimal incubation time was found to be 60 minutes, that is, in lane 6. If we only use 50 minutes of incubation time, then we would still see the 2700 base pair undigested plasmid. Hence, 60 minutes is the shortest time needed in order to digest the plasmid into its fragments. Now that we've determined the optimal conditions, we look towards determining the RE site. We performed AGE again with the 1000 base pair DNA standard in lane 2, as well as the digested DNA standard with REX at the optimal condition set seen in lane 3. By establishing a standard curve for the DNA ladder with distance in arbitrary units against the logarithm of the molecular weight, we get a linear equation which we can use to estimate the fragment sizes in lane 3. The estimated fragments are 590, 242, and 167 base pairs. These fragments could be arranged in many possible combinations. Hence, the cut site of REX will depend on which combination shows a common cut site between the fragments. Upon checking each combination, we found that the combination of fragments is 167 base pairs, followed by 242 and 590 base pairs. With these fragments, the common cut site, as seen on the figures on the left, is NGTACN, where N is any nucleotide. So at 167 base pairs, seen at the top figure, we see TGTACT, while at 167 plus 242 equals 409 base pairs, we see GGTACC.
Now let's go into Activity 6, Part 1, Guide Questions A to D. So for the first question, explain the concept of host-controlled restriction and modification. So the restriction modification system is a defense mechanism of cells where it's basically divided into two parts, restriction and modification. The first part, restriction, is basically the synthesis of restriction enzymes which allows the host, example the bacteria, to defend from invading agents such as bacteriophages, where the restriction enzymes cleaves the DNA of the infectious agents. On the other hand, so that the restriction enzymes of the cells won't digest the cell's own DNA, modification is also carried out. This is done through DNA methylation by metal transferase, where metal groups are added to the host DNA, specifically in the restriction sites to block the cleaving action of the enzymes. The second question is, what are the usual properties of recognition sites of type 1, 2, and 3 restriction enzymes? So for type 1, the recognition sites, it's usually bipartite, which means it has two parts. So for example, is EcoK1. For its first part, it has a 3 nucleotide long site, which is AAC. Then for the second part, it has a 4 nucleotide long, which is GTGC. Then it's usually asymmetric as shown. Then for the second type, it's the most common and it's usually 4 to 6 BP long and it's usually palindromic. So for example, is the EcoR1, which is GAATTC, and its reverse complement is the same, which is GAATTC. Then for the third type, it's usually 5 to 7 base pairs long and usually asymmetric. So for example, is EcoP1 and its restriction site is AGACC. For the third question, enumerate some of the factors that may affect restriction enzyme activity. So first will be the temperature, where most have an optimal temperature of 37 degrees Celsius. Second is the buffer system, where the optimal pH is usually around 7 to 8. And another is the cofactors and ionic conditions, where restriction enzymes usually require magnesium ions or sodium and potassium ions for enzyme activity. And lastly is methylation, where if adenine and cytidine is methylated, then enzyme activity there's no enzyme activity. For the fourth question, what is star activity for restriction enzymes? So star activity is basically a property of restriction enzymes where under non-optimal conditions, the specificity of the enzyme's cleaving action changes, where it may act on DNA sequences with some minor differences to the original site. So for example, is EcoR1, where in optimal conditions, it recognizes and cleaves the site GAATTC, but its star activity may result in the cleavage of TAATTC or even CAATTC.
Congrats on finishing part 1 of this presentation. For part 2, we will begin with blunt end and sticky end ligation. For this part, our gene of interest is the ZS green 1 gene, which we will isolate from PMR ZS green 1 plasmid and insert into the PUC19 plasmid. A green fluorescent protein, the brightest one commercially available, ZS green 1 is used for whole cell labeling, promoter reporter studies, and controls on transfection. But how exactly do we transfer our GOI from one plasmid to another? To do this, our first step would be to amplify the target gene through PCR. Hence, we'll be synthesizing primers. Then we'll digest the receiver plasmid and perform ligation of the amplicon into the receiver vector. Our first strategy is blunt end ligation. Here, the enzyme makes a straight cut so that the resulting fragments do not have overhangs. They are called non-cohesive ends. From our source plasmid, we see that there are three blunt end restriction enzymes, and their corresponding cut sites are shown on this slide. We amplify the GOI and should end up with an amplicon size that is the size of the GOI itself, 705 base pairs. To synthesize our primers, we copy the first 40 bases from the 5' end of the gene. This is our forward primer. We do the same for the other end, but this time we get the first 40 bases from the 5' end of the other strand. In terms of the first strand, this would be its reverse complement. This is our reverse primer. Let's try to make these primers through snap gene. Select the first 40 base pairs of ZS Green 1 and click the Primers tab, then add primer. Select top strand and rename it as blunt end forward primer, then add it to template. For the reverse primer, we select the bottom strand for the last 40 base pairs of the GOI and click bottom strand. This is the blunt end reverse primer. Now that we have our primers, PCR will yield our amplicon, which is the GOI itself. Now, there are two ways to insert the amplicon into the receiver plasmid. We could either insert it through the direction as in construct 1, or have it flipped as in construct 2. Through APE, we open the sequence of the receiver plasmid. We see the common features by going to the Features tab, then annotate features using library. We go to the Enzyme tab and select SMA1, which is the blunt end restriction enzyme that we will choose to cut the plasmid. Copying the sequence of the amplicon, we paste it in the cut site, and add this as a new feature, ZS Green 1, where the feature type is gene. And then we click OK. On another window, we insert the reverse complement of the gene, and add it as a feature once again. Now that we have our two constructs, let us open them with SnapGene and show the direction of transcription of each gene. We know from the GOI source plasmid that the ZS Green 1 gene is read from the 5' to 3' end, starting with ATGG. Hence, we double-click this and put the direction as to the right. Going back to the map for construct 1, the direction of the GOI is opposite to the lock operon, which means it will not be transcribed in the correct direction. For construct 2, it is the reverse complement, so we double-click it and put the direction as to the left. Looking at the map, we see that the lock operon and the GOI are in the same direction, meaning that construct 2 is the correct transcription direction of the GOI. By performing RE digestion and AGE, we can also determine the correct plasmid construct. We choose a restriction enzyme that has two cut sites, one that cuts within the GOI and another that cuts outside of it. By performing AGE, we expect to see different fragment sizes with each construct. In this example, construct 1 will generate a longer fragment, while construct 2 will produce a shorter fragment. Let's look for one cut site within the GOI that also cuts outside of it. This condition is met by SAL1. So let's choose construct 2 is the expected correct transcription direction. What fragments are we expecting? We have sequences of 877 and 1134 base pairs. From here, calculating the fragments as how we did in activity 6.1b, we should expect 257 and 3134 base pair fragments. We go to APE and highlight this enzyme through the enzyme selector. Making sure the plasmid is circular, we click digestion and see that the fragments are indeed as expected. Thus, this confirms that construct 2 is the correct plasmid construct. If we try to digest construct 1 with SAL1, we see different fragment sizes at 482 and 2909 base pairs, which are not the expected fragments.
Now, instead of making blunt ends, this time we're going to do sticky end ligation. To do this, we need to first synthesize a primer that allows us to place restriction sites that produces sticky ends on both sides. Then we digest both the PMR ZS Green 1 plasmid and the PUC19 plasmid and perform ligation. Our first goal is to synthesize a primer, but before that, we have to know what amplicon we want to achieve so that we'll be able to get an idea on how to construct the primer. So for the amplicon, what we want is that the ZS Green 1 gene, our gene of interest, to be flanked with restriction sites on each side, where for this virtual experiment, we're going to use XBA1 as our sticky end producing restriction enzyme. We also want that the restriction sites to be directly beside the gene of interest so that when we perform digestion, we just get this. As you can see, our gene of interest will be flanked with sticky ends if we digest the amplicon. Since we now know our desired amplicon, we can now design our primers. So for our forward primer, we want it to have 30 base pairs from the 5 end of the top strand. Then the other 10 base pairs will be for the restriction site. The same will be done for the reverse, but instead of the top strand, we'll copy the 5 end of the bottom strand. With that, let's try to construct our primers in the SnapGene software. So first, we open the PMR ZS Green 1.FASTA. Then let's find the ZS Green 1 gene and add the first 30 base pairs of the forward primer. And let's name it forward primer. Then let's find the XBA1 site and copy 10 base pairs. And let's add it to the forward primer. Next, we add the reverse primer. So let's copy another 30 base pairs again and name it reverse primer. And let's go back to the XBA1 site and copy another 10 base pairs. Then while we're at it, let's also make the amplicon file. First, we open the APE software. Then we copy the first 10 DNA sequence, then the gene itself, then the last 10 DNA sequence. However, we need to note that we have to get the reverse complementary since what we want is the top strand. So since our gene of interest is 700 base pairs long and we extended it with 10 base pairs on each side, then the amplicon side is 725 base pairs. And lastly, let's check if the two XBA1 sites are present by opening it in SnapGene Viewer. So as you can see, we have two XBA1 sites. So here's the summary of what we got. Here's the DNA sequence for the forward primer and the reverse primer and the size of the amplicon. Now that we've prepared our gene of interest, let's look into our vector. So at around the multiple cloning site of the vector, we can also see a XBA1 restriction site. And so with digestion, we'll also get the same sticky ends as our gene of interest. We can now perform ligation, where as a result, we'll get our first plasmid construct. However, it is important to note that since the restriction site is palindromic, then if you rotate the gene 180 degrees, it can also ligate with the vector as shown in the screen. So as a result, you'll get two different constructs in this activity. Now let's do these constructs in the APE software. For the first plasmid construct, first we open the amplicon file on SnapGene. Then we copy the top strand of the amplicon from the gene of interest to one of the restriction site. Then we open the PUC19.FASTA file and paste the strand beside the XBA1 site. Then we add the gene as a new feature, name it as ZS-green1, and label it as gene. That's your first construct. Now let's look at the plasmid map of construct 1 by opening the file on SnapGene and adding the common features. We do the same for the second plasmid construct, except that we take the reverse complement of the gene of interest. So we do the same, we add the new feature, name it as ZS-green1, and label it as a gene. Then we open it in the SnapGene viewer and add common features. So here's our second plasmid construct. Now we have obtained the DNA sequences of the two constructs made from the sticky end ligation, but there's only one correct construct, and that will be construct 2, since the transcription direction of the ZS-green1 gene follows the direction of the other feature, especially that of the lac promoter. We can also differentiate the two plasmids through RE digestion, just like in blunt ligation activity. For this one, we use STI1 and SAP1 restriction enzymes, where the STI1 is found in the gene of interest. Now let's go to activity 6, part 3, RE digestion as a powerful screening tool. This is another activity to test out whether you've understood how to make virtual digest and how to determine the right construct with the resulting gel profiles. So in this experiment, we first do a sticky end ligation where the target gene, gene Z, was inserted in the multiple cloning site of a plasmid vector. Then we do bacterial transformation and growth, where 5 E. coli colonies that might have taken up the plasmid with gene Z were grown in lysogeny broth with the appropriate antibiotic. Then the plasmids were isolated after an overnight shaking incubation. With the DNA sequencing of the 5 isolated plasmids, we've obtained 5.FASTA files, which we could use for virtual digestion. Now we are left with a question: which colony contains the correct construct? As discussed in the previous activity, just a simple digestion and gel profile comparison could tell us which plasmid had the correct insertion of gene Z. So first, the correct insertion of the gene Z was initially analyzed using the APE software, and according to it, RE digestion with HIN3 showed result in 3 DNA fragments of sizes 3801, 2623, and 1300. With this, we could do virtual digestion and virtual gel electrophoresis with the 5 DNA sequences we got, and the one with the same resulting DNA fragments will then be the correct construct. Let's go through again on how to perform enzyme digestion using APE software. So we just have to press digestion dialog, and there you have it. Your gel profile and your DNA fragment sizes. So after analyzing each of the plasmids using the APE software, we'll obtain 5 digests. Now let's check which one is the correct construct. For plasmid 1, if you look at the table on the upper left, you can see the DNA fragment sizes. So comparing that to the theoretical DNA fragment sizes, it seems that plasmid 1 is not the right construct, since the second fragment has a different size. The same is true for plasmid 2, where only two fragments matched, and plasmid 3 then. Now for plasmid 4, it seems that this is the correct construct since all of the DNA fragments matched. We can also observe that even without DNA sequencing, we can determine which construct is right just by doing RE digestion and age. You just have to pick an appropriate restriction enzyme or enzymes, which could help you differentiate different plasmid constructs. But let's also check the last plasmid to be sure. And just like the previous, it seems that there are only two same-sized fragments. For the second to the last activity, 6.2 part 4, sticky and ligation with two restriction enzymes. In this activity, we're going to now try using two restriction enzymes instead of one to do sticky and ligation. Here, we're going to use SAL1 and BAM-H1. For our first amplicon, we'll be making a BAM-H1 forward primer and a SAL1 reverse primer. And below, you can see the sequences for each primer. The same is done for the second amplicon, except that the forward primer contains SAL1 site, while the reverse primer contains the BAM-H1 site. Now that we've prepared our amplicons, let's look into our vector P flag CMV2 plasmid. So around the multiple cloning site of the vector, we can see both SAL1 and BAM-H1 restriction sites. And so with digestion, we'll also get the same sticky ends as our gene of interest. But note that the sticky ends made from BAM-H1 can only ligate with the same sticky ends. So for amplicon 1, the reverse complement must be the one inserted into the vector, where this construct 1 will be the result after ligation. On the other hand, for amplicon 2, there's no need to get a reverse complement since the SAL1 sticky ends are both found on the left side. So we can easily insert the gene of interest as shown. This is our construct 2. Just like the previous activities, there's only one correct construct, and that's construct 2, as it contains the correct transcription direction. Specifically, construct 2 is the correct construct since the CMV promoter has the same direction with the gene of interest, and that the promoter is found upstream of the gene of interest. Again, similar to the previous activity, restriction enzyme digestion can be used to determine the correct construct. For here, we use BSRG1 to digest the plasmids. And if the experimental gel profile is the same with the gel profile for construct 2, then that plasmid is the correct construct. So for our last activity, which is activity 6 part 1, guide questions E to F, for the first question, what are other alternative methods to genomic DNA? So one is TA cloning, which will be discussed in a later slide. Second is gateway cloning, which uses two enzymes called the BP clonase and LR clonase, where BP clonase is the one that makes an entry clone, while LR clonase transfers the gene of interest from the entry clone into an expression vector. The third would be infusion cloning, where the gene of interest is basically extended with around 15 base pairs that is complementary to the vector ends. Then this amplicons is then annealed to the linear vector. For the last question, what is the principle behind TA cloning? So the TA cloning technique takes advantage of the fact that adenine and thymine bases do pair with each other, and that some polymerase, such as Taq polymerase, leaves a 3 and A overhangs to PCR products. With this, the gene of interest can be ligated to a linear vector that contains the complementary overhang, which is the T overhang. So for our conclusions, first is SnapGene Viewer allowed us to view our plasmids and see its future. And with this, we were able to read plasmid maps. While APE software allowed us to edit the plasmids and make virtual digestions. Then digestion for inserting gene of interest can also be done with one or two restriction enzymes, and ligation can be done with blunt ends or sticky ends. It is also seen that the correct plasmid construct can be determined using not only DNA sequencing but also through RE digestion. Now here's our recommendation. First will be to perform actual gene insertion, RE digestion, and gel electrophoresis in lab. And second will be to perform virtual TA cloning and other cloning methods. Here's our references. Thank you for listening.